Buenos días a todos. La charla es en inglés, pero después podemos hacer la discusión como vosotros queráis. So today I'm going to talk about my project that focuses on the epigenetic basis of the interactions between the mosquito vector and its malaria parasite in the case of human malaria. So to start, I would like to remember you what is epigenetics. Epigenetics refers to the changes in the phenotype or gene expression that are not caused by changes at the level of the DNA sequence. There are several mechanisms of epigenetic change. We have DNA methylation, that is the addition of a methyl group to the DNA chain. There is also modifications of the tails of histone proteins that form these nucleosomes, that is the octamer of histones along which the DNA is wrapped. And then this level, they can be further structured into loops that at the end form the different chromosomes. So all these different mechanisms affect gene expression. For example, in the case of histones, typically the acetylation, the addition of a acetyl group to the tail of histones will cause the chromatin to open, and then this will favor gene expression, whereas the addition of a methyl group will promote packaging. And then we have the chromatin close, and this is typically associated with gene silencing. So my research line focuses on the epigenetics of host-parasite interactions, but why is important epigenetics in this context, in the context of host-parasite systems? Host-parasite systems are among the most dynamic and plastic systems in nature. So adaptations between the parasite and the host occur very quickly. And in this sense, epigenetic mechanism provides a source of variation that can be rapidly shaped by host and parasite selection pressures. So my question, the question that I aim to address in my investigations is to which extent the phenotypic variation and the plasticity that occur during the interactions between these two guys are mediated by epigenetic mechanisms. And I aim to address this question in the context of malaria disease. The term malaria comes from the medieval Italian bad air that so the origin of the name is because the disease was associated to the air that people breathe in the swamps or marshlands. And before the parasite was discovered, was thought that was like caused by this, by the air in these swamps and marshlands. So, but now today we know that malaria is caused by parasites of the genus Plasmodium that are transmitted by Anopheles mosquitoes. And in the case of human malaria, the most prevalent and morbid species that cause human malaria is Plasmodium falciparum. It is still causing more than 500,000 deaths a year, and it's also a major cause of poverty, mainly in African countries. At present, there is no effective vaccine, and this is mainly because the interactions between the mosquito and the parasites are very context dependent and are influenced by a number of environmental factors. So this is the malaria life cycle. So a mosquito that is infected with the disease will inject these guys that are the sporozoites, is one of the transmissible stages. These sporozoites go to the liver and infect liver cells. They undergo a number of replication sexually and release this other form that are called merozoites to the bloodstream. In the bloodstream, these merozoites infect red blood cells. And here it starts this intraerythrocytic cycle that goes through different stages, ring, trophozoites, and chrysomes. The chrysomes eventually, under different environmental conditions, will differentiate into gametocytes, that is the sexual form. These gametocytes then is the other transmissible stage and is taken by another mosquito when another mosquito bites this infected human. And then starts the cycle in the mosquito vector. These gametocytes differentiate into gametes that fusion and form the zygote. This zygote converts to a motile form that goes to the midgut epithelium of the mosquito, and then this forms a kind of cyst that is called oocyst, and there is a lot of multiplication here, and differentiate into the sporozoites that go to the salivary glands and then close the cycle again. So what we know about gene regulation and malaria studies thus far has mainly focused on this part of the life cycle of the parasite in the red blood cells and in culture conditions, so culture parasites. But my research focused on the life cycle of the parasite in the mosquito, which in terms of gene regulation and more specifically in terms of epigenetics, remain a black box. And this is despite of the implications of this cycle for malaria epidemiology and control, because actually the mosquito stages are nowadays the main target for malaria control interventions. So before I mentioned that the interactions between the parasite and the mosquito are very context dependent, so it's very important to study the disease in the natural context of transmission. And this is why this project takes place in a malaria endemic area in Africa, in Burkina Faso. And here almost all the population is infected with the parasite at some point in the year. So we went there, I was there, and then we screened children in school for the presence of the parasite in the blood, in blood smears. Those that had the parasite at high prevalences were asked to be participants in the study. We took blood, and with this blood, with this kind of thing, we we infected experimentally mosquitoes. So the blood is disposed in this valve, and then is through a membrane, a parafilm, it's put in contact with the mosquitoes, and the mosquitoes get crazy to feed them on this, and then the mosquitoes get infected. This is a schematic, the same thing schematically. We took infected blood and also control blood, in which we kill the parasite by temperature. So this is a real negative control, and then with this we infect the mosquitoes. Then these mosquitoes that were infected, we performed dissections at different time points that correspond to the different developmental stages of the parasite in the mosquito. Time 0 before infecting the mosquitoes, then at time 7 days that correspond to the oocyst here in the midgut epithelium. This is a midgut. And then at day 14 in the salivary glands, which is the sporozoite stage. I want you to remember these figures here and these names. And then on these tissues, we perform transcriptomic analysis, total RNA and small RNA, and then ChIP-seq. For those that don't know, the ChIP-seq is a technique that allows you to study histone modifications in the DNA. So if you have, this is a DNA wrapped around nucleosomes, and these little balls are the modifications, either acetylation or methylation or whatever. Then what you do is that you use these antibodies that, in this case, well, this is a protein, but it would be this case. In this case, they bind to a specific histone modification. And then you will isolate only those fragments that have this particular histone modification. And this is what you see here. So you see the genome with gene 1, gene 2, and then you have a peak of this particular histone here, meaning that in this region there is a lot of this. Okay, so that's more or less what that means. And then what we aim to see based on these profiles is that for a particular gene that probably, so we studied like 3 different histone modifications that are associated to active transcription. They are going to be abundant in these regions that are being transcribed. And then one histone modification, this one, that is associated to genes that are silent. Okay. So this approach allows us to simultaneously study the parasite and the vector during a malaria infection. Now I'm going to present first the results of the parasite, so to which extent epigenetic mechanism regulates the phenotypic plasticity of the parasite in the mosquito. What we know about phenotypic plasticity of the parasite is in the blood stages, as I said, and in these stages and in cultural conditions, we know that the adaptation ability of the parasite to respond to the conditions that encounters in the host depends on these families of genes that are called clonally variant genes. So these genes have the particularity that they are encoded by multiple genes. So multiple genes encode the same protein, and then the parasite switches between different copies, then expressing different variants of the protein. And this way, for example, in the case of antigenic variation, they are able to evade the immune system of the host. One of these families is the bar gene family, that it's so is this protein PFM1 that is encoded by 60 different copies of these bar genes, and this the parasite expresses this protein. This is a parasite. This is a red blood cell. The parasite produces a protein, and the protein goes to the surface. In the surface, it mediates the adhesion of this infected red blood cell to the endothelial cells, and then this infected erythrocyte is not cleared by the immune system of the host in the spleen. So this is a real image of an infected red blood cell, and these little dots here are these proteins at the surface of these infected erythrocytes. So what we know in the blood stages and in vitro is that every infection cycle, the parasite is able to switch between these different variants for this protein, and this is what is called mutually exclusive expression. So the parasite expresses only one gene of all 60 possibilities, and this is mediated or is associated with epigenetic changes. As I say, this is for the blood stages, but we don't know what happened in the case of these clonally gene families in the mosquito during the life cycle of the parasite in the mosquito. So imagine that these are all 60 copies of the bar genes. In the late forms of the parasite in the blood stages, only one is expressed, and then there is this immune evasion that I just talked about. And then what happened in the mosquito? So it may be that the same form is maintained through remember that here there is sexual reproduction. So in theory, all epigenetic marks and everything should be erased. So it may be that the same variant is maintained, so there is a epigenetic memory, that there is a reset, and then all variants are expressed, so the parasite is somehow prepared to whatever it finds in the next human host. Or it can be that there is a reset, all variants are silent, and then a new variant is expressed, which is different from the one that was expressed in the human host. This has a tremendous impact at the level of epidemiology and therapeutics, because the parasite is somehow in this case the parasite, this could be a response to the selection by the human host, because the parasite is adapting to the human host population, whereas in this case this would be mediated by selection by the mosquito vector. So these different possibilities is what we are trying to answer in this project. Now, but the results is that what we found is that in the mosquito, a large number of these clonally variant genes are expressed. This is I'm going to explain this figure. This is the oocyst, and this is the sporocyte. This is midgut, and this is salivary glands. This stage is the one that is transmitted to the next host. So what we see is that somehow most of these genes are silent, and in the sporozoite there is upregulation, so overexpression of these genes prior transmission to the next host. this is the expression, overexpression in one side, overexpression in the other side, and this is the significance of these changes. Among these genes that are expressed at high levels in the salivary glands here, there is a number of transcription factors that in the case of Plasmodium, now we know that now we are starting to discover that play important roles in the regulation of gene function, but there is still very little knowledge about it. So what we see in the mosquito is that there are a number of these transcription factors. Each row, okay, this figure is represents the expression level for each stage. So this is the blood, different replicates, the oocyst, and the sporozoite. So, for example, this group of genes here will be highly expressed in the sporozoite and less expressed in the oocyst. Okay, and the contrary, for example, here, these ones are very highly expressed in the blood and less expressed in the oocyst and highly expressed in the sporozoite. So by this way, we identify this factor that is highly expressed in the sporozoite, and we also found that this factor, this particular transcription factor, has binding sites. This is by computational analysis. Has binding sites in the genes that are also highly expressed in the sporozoite. These clonally genes that I showed before that are very highly expressed. So these genes have binding sites for the transcription factor, suggesting that somehow this transcription factor is produced, then binds to this DNA sequence motif, and then activates the transcriptional cascade that we see in the sporozoite. Apart, so about the case of the bar genes that I explained you before, as I said, in the blood stages there is only one bar gene that is active, but we didn't know what happened in the mosquito. In the mosquito we found also a bar gene that only one bar gene is active, and this was not active in the blood, and is first a little transcribed in the oocyst and then reaches the highest level of expression in the sporozoite prior transmission. We did this for different replicates, for different infection replicates, and in all cases we see the same pattern. We also show that associated this active bar gene has this binding site, this DNA sequence that corresponds to the binding site for this transcription factor that I showed you before, that is highly expressed in the sporozoite stages in the salivary glands. And there is also transcription of a long non-coding RNA, which other studies have shown that has an enhancer activity, so promotes also expression. None of these two things happen in the other copies of the bar genes that are not being expressed. Okay, so what we would conclude from these results is that the model that fits our results is this one. So there is a reset and then switch to a new variant, and that this would be driven by the selection, by the interaction between the mosquito and the parasite. How that is possible? Well, what we propose is that probably this protein may be functioning in the mosquito in a similar way that it does in the blood stages of the parasite. So somehow mediating the adhesion of the parasite to the endothelial cells. But this is something that we still need to investigate, because we don't know which tissue it happened and which stage, and whether it holds true or not. Now I'm going to present the results on the mosquito side. So in the mosquito we know that a malaria infection causes a number of changes at the level of the phenotype, and this affects life history traits such as longevity and fecundity, and also affects vector competencies. But we don't know about the mechanism that regulate these phenotypic changes. in a previous studies, we show the link between changes at the level of histone modification patterns and gene expression, and that the patterns were similar as those that have been described for Drosophila. Somehow, this was a proof of concept about this relationship, which is important now to study this, but in the context of a malaria infection. And this is what we did. So we compare histone modification profiles and gene expression between infected and control mosquitoes. So this is, for example, if there is this is what we are looking for. So changes, for example, this is a gene, this is the promoter, and then in non-infected we would have this pattern. If this gene is silent, so a lot of this silencing histone mark, and then if this gene is turned on, then we see an increase in other marks that are typically associated to active transcription. So this was the type of pattern that we were looking for. And these circles, what they represent is that the number of regions that are different between infected and non-infected. The overlapping is the regions that are not significantly different. These are significantly different and higher in infected. These are significantly different and higher in non-infected. And this for each histone modification mark. What we also found is that this region that we call differential histone modified regions associate to genes that are also differentially expressed. And some of these genes turn to be genes that are involved in mosquito immunity. Now, the next question is, okay, is there something in these genes that they have something in common in the promoter region? So, for example, sites for transcription factors, or are these genes enriched in a particular sequence that may be an indication that they are regulated by the same mechanisms? And the question is that, yes, we found particularly interesting was the fact that we found these binding sites for these two transcription factors, FOS and JAM, that in the mosquito has been recently suggested to participate in these signaling pathways that are associated to immune responses. So this is an early finding that we still need to work on, but I think that was really interesting. Then, a part of histone modifications, small non-coding RNAs can also participate in the regulation of transcription. In particular, in the mosquito, microRNAs play an important role. So if you don't know much about it, so these microRNAs are produced in the nucleus. They are processed in the cytoplasm, and in the cytoplasm they bind this, or they are capped by this protein complex that is called RISC. And this protein complex takes them to the messenger RNA. This is a messenger RNA to which these microRNAs are complementary. So each microRNA is complementary to the 5' region of the target gene. And this is going to cause either so this messenger RNA is not translated into proteins, or they cause degradation of the messenger RNA. So in the case of the mosquito, what we see is that a number of, there are all these microRNAs that have been previously described for Anopheles gambiae, are differentially regulated, are different between infected and control. Infected is red, control is blue. And then this is the same rationale that I explained before for the other heat map. And then what we also see is that if all these microRNAs that are differentially expressed, these are based on computational analysis also. These bind to all these target genes. Okay, each dot is a target gene, and this is a microRNA. So these microRNAs has binding sites in all these target genes. And for all these target genes that are predicted to bind these microRNAs, there are a number of genes that also participate in immune functions. Therefore, suggesting that the differential regulation of microRNAs also regulates responses to malaria infection in the mosquito host. Now, as a summary of the two parts that I just talked about, in the first case, in the case of the parasite, we showed that the passage through the mosquito impact the regulation of clonally variant genes in Plasmodium falciparum, and that a single gene is active, and this is probably caused by the selection by the host immune system. In the case of the mosquito, we saw that we could identify malaria responsive epigenes, so genes that show changes at the level of histone marks or microRNAs and gene expression. And we could also identify the binding sites for transcription factors that could target genes linked to immunity. Now, this is related to what we previously did. Now, what the ongoing work that I'm at this moment working on is particularly in the parasite, we have the hypothesis that the regulation of this clonally variant gene expression in the mosquito is driven by transcription factors. So, to study this transcription factor regulation, we aim to perform this type of technique that is called ATAC-seq. This technique is based on the use of a transposase of a gene that is going to cut. These are nucleosomes. Nucleosomes, the DNA group around the nucleosomes, and then these are several nucleosomes in a region of very packed chromatin. Okay, so this transposase that is here, that is linked to adapters, is going to cut all the regions that are nucleosome free, so they don't have nucleosomes and they are open. Okay, so they are going to cut all these regions. They are also, so there are two types of regions that are going to escape to this cutting. Regions where there are nucleosomes, and regions where there is a transcription factor bound here. To imagine that here you have a protein, and then the fragment that is linked to this protein is going to escape of this cutting. So we will have information about the position of these nucleosomes, and also the presence of a transcription factor binding sites. So this is what we aim to do, because these have not been performed in Plasmodium yet. And as I mentioned, transcription factors are still something that is under study in the case of Plasmodium falciparum. So we are performing this in collaboration with Alfred Cortés in Barcelona from the IS Global, and with José Luis Gómez-Díaz here in Sevilla at the CAP. And then this fall, I'm doing the same thing, but in the context of the life cycle of the parasite in the mosquito. And I'm doing this in the context of a collaboration with Lisa Cartwright at Glasgow University. And then also something that we will perform next year will be the comparison, because as I said, the other hypothesis that we have is that this the regulation of these clonally variant genes is mosquito driven. So we aim to compare infections in different mosquito vectors. So gambiae, colucci, which is a different species, and then arabiensis, which is farther away, to see to which extent the patterns that we see in Anopheles gambiae are different in these two other mosquito species. And this we will do it in Burkina Faso in collaboration with Thierry and Serge. And then something that is very recent, because that happened last week in the conference in International Entomological Conference in Orlando, is a new collaboration with Carolina Varillas-Muri from the NIH in Washington. They have characterized, or they are characterizing some of these proteins that are involved in the immune response of the mosquito to the parasite, and they have evidence, or they have the hypothesis that after an infection, there is an epigenetic priming, so that the mosquito gets primed. Somehow, some of the proteins, by some epigenetic mechanism, remember the infections, and this will affect the next infection in the mosquito. So we are going to perform epigenetic analysis in this, in the context of their investigations. And that's all what I have to say today. So my thanks to my previous lab at Emory University. This is Victor Corses, that gave me the opportunity to start this project and believe that there was some good on this. And then people in Burkina, Serge and Thierry and all the technicians, and then all these people that participated in the study. And thank you very much.